Yeah.
Espero que arribi tot el geni.
Tanquem la ràbia.
Aquesta no.
Es pot encendre una llum allà.
Sí, aquí darrere la columna.
Sobre la sessió, anem a procedir a la defensa de la memòria que per a optar al grau de doctor en biotecnologia, doctor per la Universitat de Barcelona, presenta el senyor Marius Tomàs i Gamissat. The session will be held in English, so Marius, the floor is yours.
So thank you. Here in this presentation, I will give an introduction, then the objectives and overview, 4 chapters of results, and a final summary, and the general conclusions. Regarding the introduction, I will present Picha pastoris as a cell factory, and then give a few slides on systems metabolic engineering. So as all of you know, yeasts have been used for centuries for bread, beer, and wine production. Nowadays, they are extensively used in industrial biotechnology due to its advantageous properties, such as the high growth rates, the ability to grow up to high cell densities, and to perform post-translational modifications. Among yeasts, the methylotrophic Picha pastoris has become one of the most extensively used eukaryotic systems for recombinant protein production, especially those that need to be secreted to the cultivation media. When producing recombinant proteins, we could have some bottlenecks, and these 2 main bottlenecks are the protein folding and secretion processes and the metabolic burden associated to the additional building blocks and energy demands. In order to overcome these limitations, we can find 2 different kinds of strategies. The first one is the process engineering strategies, and for example, we can use cross-substrates, provide amino acid or vitamins or precursors to the cultivation media, or modulating the environmental cultivation conditions, for example, using restricted oxygen availability. On the other hand, we also have cell engineering strategies. These strategies were first used based on the pre-existing knowledge on cell physiology and biochemical pathways. Then, the second generation strategies arose from the development of different omic techniques. Then the third generation of strategies were based on systems metabolic engineering. So now the question is, what is systems metabolic engineering? Basically, is the integration of the classical metabolic engineering aiming at a rational strain design combined with the physiological analysis from a systems biology perspective. All this generated data can be integrated into models to close the systems metabolic engineering cycle and then predicting new targets for strain improvement. So, as I said, systems metabolic engineering is closely related with systems biology. That is the discipline that tries to describe the biological systems as a sum of multiple layers from genomics or from the genotype up to the phenotype. Each one of these layers can be characterized by a specific set of omic tools. Regarding Picha pastoris, in the last 10-15 years, several omic platforms have been developed and implemented for disease. For example, we have three public genome sequence available. In addition, there's several data on characterization of gene expression in different conditions, the same for proteomics, lipidomics, and especially, in particular, our group has been involved in developing, adapting, and implementing metabolomic and fluxomics analytical techniques. So these techniques are based on using a label substrate and then applying some calculations in order to elucidate the metabolic flux distribution. The first method to be used in Picha pastoris was the metabolic flux ratio analysis that basically establishes the ratios between converging metabolic pathways. So using these ratios as constraints, the flux balance analysis can be performed. This method has been successfully applied for cells growing on glucose and mixtures of glucose or methanol. In addition, it was also tried for glycerol and methanol. Nevertheless, the reduced number of carbon atoms of glycerol made that the information available or the constraints were not enough for performing the flux balance analysis. More recently, another strategy has been implemented, that is the iterative fitting of both mass-isotope distribution together with extracellular fluxes. In this case, it has been applied for mixtures of glycerol and methanol and glucose on methanol, both in stationary and instationary cultivations. All these methods usually use central carbon metabolism network. Nevertheless, flux balance analysis can also be performed at a larger scale, that is using the genome scale metabolic models. The genome scale metabolic models are basically, or they contain the whole metabolic network of an organism. In order to develop them, we need information from the genome. Then we also take information from databases, biochemistry, and physiology. And then we gather this information together in order to establish the gene-protein reaction association. With this information, together with biomass composition, we can formulate a metabolic network that is represented as a stoichiometric matrix. Then this network has to be validated with experimental data. In addition, the integrative analysis of this experimental data can be used for model refinement and subsequent implementations. But which are the main applications of genome scale metabolic models? So the most obvious maybe is that is a huge database that contains genes, proteins, reactions, metabolites. In addition, we can use them for contextualizing high throughput data. For example, we have large amounts of information that maybe are meaningless for us, but putting this information on a map, maybe we can figure out what's going on with our data. In addition, it integrates multi-omics data, for example, from the genomics up to the fluxomics. Maybe the most well-known application for genome scale metabolic models is to guide for metabolic engineering. So using these models, we can predict new targets for strain improvement. In addition, and we will see in one of the chapters of results, we can use the models for assisting in biological data interpretation. Moreover, we can use the models for discovering network properties such as pathway redundancy, alternative pathways, and so on. And finally, we can also use them for analyzing the multi-species relationships by comparing the network topology. In the case of Picha pastoris, there were 3 different genome scale metabolic models available previous to this thesis. Now the objectives and overview. First of all, I would like to introduce the research group in which I performed the thesis. So our group aims at cell engineering Picha pastoris for recombinant protein production. In addition, in the context of an European project, it also aimed at producing newer strains for high value-added chemical production. Therefore, our group has provided extensive information cultivation datasets. In addition, preceding theses had broadened the body of knowledge on systems biology. This information was used for designing newer strains. Nevertheless, there's still a gap here that we will try to fill in this thesis, that is using systems metabolic engineering as strategies for cell engineering. So the aim or the objective of this thesis is developing an integrated genome-scale metabolic model by merging the pre-existing ones and then extending its capabilities for new carbon sources. In addition, we want to prove the model effectiveness by applying it on two different systems metabolic engineering applications, that is metabolic flux analysis and to help to assist biological data interpretation. Here as a graphical overview of the work done in the thesis and also the chapters of results that I will present now. So in the first chapter, we merged the three available metabolic models into a single one, and then we upgraded and adapted for both on glucose and mixtures of glycerol and methanol. In the second chapter of results, we extend this model to be able to predict growth on glycerol and methanol. And finally, in the last two chapters of results, I will present the two applications, that is the metabolic flux analysis and assisting to the physiological data interpretation. So the first chapter of results is the generation of a consensus genome-scale metabolic model for Picha pastoris. As I said in the introduction, there were available three different genome-scale metabolic models previously to this thesis. Nevertheless, they seem to be reconstructed in a semi-automatic way, as there's missing or diverging or contradictory information among them. For instance, misannotations in public databases, if they are not created in order to incorporate this information into the models, we can have wrong gene-protein reaction associations. In addition, all of them were based on a Saccharomyces cerevisiae model as a scaffold. Therefore, some Picha-specific traits were missing. So the objectives of this third chapter is to provide a reliable genome-scale metabolic model, and we will perform this by merging the existing models, curating the resulting draft, and extending our network for these Picha-specific pathways as well as new biomass equations. Here's a graphical representation of the experimental procedure. We first merged these models into a consensus draft, then we upgraded the resulting model, and finally we validated. A little bit more in detail, the workflow that we follow for developing this model. The first step was metabolite identification and reunifying. Each one of the models used a different reference database in order to build or to take the metabolite names or IDs, so they were different in all the models. We had to homogenize this nomenclature. In addition, we create some gross errors that we found in the models. In the first step of merging, we just paired the exactly equivalent reactions. In a subsequent step, we then compare the rest of reactions by checking the reaction reversibility, stoichiometry, gene associations, and complementalization. Finally, when we had this initial reconstruction draft, we then analyzed those specific Picha pastoris pathways that were represented in the model, and then extend the model by incorporating new reactions to represent such pathways. Finally, we incorporate new biomass equations and validate our model. Here is a summary of those Picha-specific pathways that we updated. For example, the fatty acid metabolism, there were really contradictory information regarding its compartmentalization. In addition, we find several differences with respect to Saccharomyces cerevisiae in the oxidative phosphorylation, glycosylation pathways, as well as sphingolipid metabolism. And of course, the specific biomass composition for Picha. So in the end, we had a reconstruction with a large number of genes, metabolites, and reactions. Indeed, so that means that we have a more extended and detailed model. Indeed, we incorporated almost 200 new reactions to the model. So this was a reconstruction, but what we really want is to build a model, so models are able to reproduce experimental data. So we really had to test if we are able of doing that. So we tried with two different data sets from chemostat cultivations. That is, cultivations performed in glucose at different oxygen availability conditions and both non-expressing and expressing an antibody fragment. In addition, we also compare the model performance with cultivations using mixtures of glycerol and methanol, using different mixtures of glycerol and methanol at two different growth rates. As you can see here in black bars, our model is able to accurately reproduce the major macroscopic growing parameters, that is, the specific biomass production rate as well as specific CO2 production rate and oxygen uptake rates. Okay, so our model is able to reproduce such experimental data, but now the question is, was it worthy? I mean, there were already 3 available genome-scale metabolic models, so that was our work improved the previous one. So we decided to check it. And for example, here is a representation of the specific biomass production, both in glucose cultivations at different oxygen availabilities, as well as the mixtures of glycerol and methanol at different growth rates. As you can see here in red, our model can more accurately predict the experimental data sets in comparison to the previous models that are represented in green. So, in summary of this first chapter, we performed the model merging. Then, with the resulting draft, we increased the number of gene-protein reaction associations and realizing a more extended and detailed model with Picha-specific metabolic pathways, including new biomass equations. Finally, we validate this model in 2 sets of different conditions, showing greater accuracy than the previous models. So the second chapter of results is benchmarking IMT 2026 for growth on glycerol or methanol as sole carbon sources. So the first question is, why glycerol and methanol? Well, in the case of glycerol, it is produced from as a byproduct in biodiesel production, and methanol can be easily synthesized from biomass after a syngas process. So they are cheap compounds that can be used as substrate for recombinant protein production and production of high value added chemical. So we can revalorize this compound for interesting applications. The previous models contain a unique biomass equation that we incorporated in our model, and in addition we extended with a new one. According to an interesting article published in Metabolomics, an accurate representation of biomass is really important in order to perform good predictions of our model. So, if we wanted to use both glycerol and methanol in our model, we had to update this model by incorporating these 2 new biomass equations. So the objective of this chapter is to extend IMT 2026 capabilities to accurately predict the described growth on methanol and glycerol as sole carbon sources. In order to do that, we perform a series of chemostat cultivations at different growth rates by using glycerol or methanol as sole carbon sources. We took samples of these cultivations and then characterized the biomass. And finally, we integrate this data into the model. Here is a representation of the main macroscopic growing parameters that are the specific substrate and CO2 production rate, as well as the oxygen consumption rate. As you can see, they follow quite a linear behavior, so there's a correlation between these parameters and the growth rate. So then, at different growth rates, we took samples of the biomass, and here's the results represented as fractions of each macromolecule for both glycerol and methanol at different growth rates. So the first we can observe here is that there's a growth rate dependent stoichiometry. If you look into the protein and RNA fraction, we can see that they are in correlation with growth rate. So at higher growth rates, we observe both for glycerol and methanol higher protein and RNA fraction. This can be explained by the growth rate hypothesis that basically says that when cells are growing at higher growth rate, they need more cell machinery. This cell machinery is commonly proteins and ribosomes. And at the same time, ribosomes are made from proteins and RNA. So it seems logical or coherent that if at higher growth rate cells need more cell machinery, and this cell machinery is protein and RNA, we can find increased content on protein and RNA. In addition, we can observe some difference when comparing compositions of cells growing on glycerol with the ones growing on methanol. As you can see here, the protein fraction of cells growing on methanol is higher than those growing on glycerol. This may be explained due to the fact that in the presence of methanol, some methanol-specific genes are overexpressed. Then this additional load of protein may be associated to these specific methanol enzymes. In addition, we also analyze the amino acid profile of the cells. And here, for example, here's a representation of the relative abundance of the amino acids. We selected just these amino acids because they are the ones that are statistically different in cells growing on methanol than those growing on glycerol. So here's the representation of cells growing on glycerol, and here the representation of cells growing on methanol. As you can see, its composition have been shifted. Now, as I said in the previous slide, when cells are growing on methanol, there's a strong induction of methanol-specific genes. So there's an additional load of specific methanol-related enzymes. So we decided to represent the composition of these methanol-specific proteins. And as you can see here in the graph, they are in the same direction respect to the glycerol composition than the methanol composition. So meaning that they are shifting the composition of cells growing on methanol towards the same, they are shifting its composition towards the same direction. So we incorporated the biomass equations into the model, and now was the time for validating the model. So we try to reproduce the experimental data that we obtained before in the chemostat cultivations. And here is again a representation of specific substrate and oxygen uptake rates, as well as CO2 production rate. As you can see here in the graph, our model can accurately describe growth on both carbon sources. Nevertheless, we find some discrepancies at the lower growth rate of cells growing on glycerol. In a recent article published last year, they performed some retentostat cultivations, and they realized that at lower growth rates, cell energy requirements are reduced. So, nevertheless, they use glucose. So we wanted to test if, in the case of glycerol, it was the same. So we had reduced energetic requirements. So here's a representation of our initial data and predictions by using the specific glycerol maintenance energy. And so we tried to reduce this maintenance energy, and what we could see is that our data gets more and more closer to the experimental represented in lines here. So in glycerol as well as in glucose, we could say that there's a reduction in the maintenance energy requirements at lower growth rates. Finally, we decided to compare our results with the previous model performance in these conditions. So we compared the deviations in the exchange fluxes from the chemostat data in glycerol and methanol. By using the previous version of the model, we get deviations for around 7 to 10%, while using the new version of the model with new biomass equations and new energetic parameters, we get deviations around 2%. Nevertheless, we also know that not always is possible to characterize the biomass in new conditions because we don't have the infrastructure or the available techniques. So we try to adjust the model without changing the biomass composition to predict these new conditions. So we use the glucose biomass macromolecular equation and then recalculate the energetic parameters according to our physiological data. Then by doing this, we reduce the deviation in the exchange fluxes to around a 2 and a 4%. Nevertheless, despite reducing these external flux deviations, an inaccurate representation of the biomass composition may lead into wrong internal metabolic flux distribution, as the stoichiometric contribution of each biomass component is different in each case. So, despite reducing or apparently reducing the deviations, for accurate predictions, we need a specific biomass compositions. So, in summary of this chapter, we perform chemostat cultivations by using glycerol and methanol as sole carbon sources at different growth rates. In addition, we characterize the biomass and show growth rate and carbon source dependent stoichiometry. We also characterize the macroscopic growing profile and showed a correlation with growth rate. Finally, we integrate this data into the model and then proceed to the model validation for both carbon sources, showing a reduction in the energetic requirements at lower growth rates and an increased accuracy with respect to the previous version. So now the third chapter of results was performed partially at the Technical University of Denmark. So we have now an extended model, and what I will show here is the first application of this model that is performing metabolic flux analysis. Previously, in our group, we attained at performing flux balance analysis by using the constraints from metaphor analysis using glycerol. Nevertheless, the reduced number of carbon atoms of glycerol together to its symmetry didn't give enough information in order to be able to perform the flux balance analysis. In addition, previous studies used metabolic networks that were derived from the pre-existing knowledge on biochemical pathways. In addition, these pathways are simplified and they don't contain a redox cofactor balance nor a specificity. So the aim of this chapter is to use the new version of the model for performing metabolic flux analysis using glycerol as sole carbon source. In order to do that, we perform a reduction of the full genome scale model to a core one. We perform some chemostat cultivations using labeled glycerol and finally compare the results with previous metaphor analysis. So here is a graphical representation of the experimental procedure. First, we took the full genome scale metabolic model and then reduced to a core one. We also performed chemostat cultivations at different growth rates with label glycerol, and finally extract, derivatize, and analyze with GCMS the proteinogenic amino acid. Finally, with all this information, we were able to perform the metabolic flux analysis. For model reduction, in order to do that, we protected some reactions that we want to keep in the core model, that they were about central carbon metabolism, amino acid biosynthetic pathways, and mitochondrial shuttles and transporters. In addition, we also protected some functions in order to ensure that our reduced model is able to predict the maximum growth rate that the full model can predict. As a result, we started from a model with more than 2,000 reactions, and we ended to a core one with just 77 reactions. Here is a representation of the physiological macroscopic parameters. There's the substrate uptake rate, the CO2 production rate, as well as the oxygen consumption rate. And as you can see, there's a good linear correlation with the dilution rate. So these results are comparable than those observed in Chapter 2 of results. So with this information and the analysis of the proteinogenic amino acid, we could then compare the results of the metabolic flux analysis with previous metaphor analysis, then try to study the impact of the dilution rate on metabolic flux distribution, and finally to check the redox cofactor regeneration and energy metabolism. In previous studies that were performed by using metaphor analysis, it was determined that there's a negligible glyoxylate pathway activity as well as a negligible activity through the malic enzyme. These observations were taken into account in subsequent studies that used the same metabolic network, and then these pathways were omitted. So what we wanted to check if our metabolic flux distribution is coherent with these previous results. Here is a representation of our distribution, and you can see here the glyoxylate cycle. There's also a negligible activity through this pathway. In addition, we also observe very low activity through the malic enzyme, so our distributions are in agreement with previous results. Unfortunately, metaphor analysis was not able to estimate the split ratio between the glycolysis and the pentose phosphate pathway. However, our methodology used here, the global fitting, was able to estimate confidence intervals for the pentose phosphate pathway. By looking to those estimations, we observed that there's higher possible relative fluxes through the pentose phosphate pathway at higher dilution rates. This is in accordance to the results shown in the previous chapter of results about the growth rate hypothesis that say that at higher growth rates we observe higher RNA content, since most of RNA precursors are generated in the pentose phosphate pathway. If at higher dilution rates we have higher RNA fractions, it seems logical to think that the pathways giving these precursors are also enhanced. In addition, we also see some growth rate dependent variations of the relative fluxes, that is, a decrease in the available relative fluxes through the TCA, and in addition, an increase in aplerotic relative fluxes, you can see here. That is also in agreement with previous results that performed the metaphor analysis by using glycerol at different growth rates and mixtures of glycerol and methanol also at different growth rates. Finally, regarding the redox cofactor regeneration, it was not used for metabolic flux analysis in previous studies. In fact, they were not present in such metabolic networks. Indeed, by performing the metabolic flux analysis, we didn't take into account this redox regeneration balance in order to avoid biased predictions. So if we haven't used for metabolic flux analysis or for determining the flux distribution, now we could use them in order to validate this or the estimated flux distribution. So what we did is to take into account the available NADH, then assuming that this NADH is giving its electrons to the electron transport chain, and then calculating the oxygen requirements both in the oxidative phosphorylation and for biomass formation. Here's the results of this estimation, and when we compare with the experimental results, we can observe that we can predict with more than 97% of accuracy these oxygen requirements. So the estimated metabolic flux distribution is consistent with redox cofactor balance and oxygen requirements. As a summary of this chapter, we performed a model reduction, then at the chemostat cultivations that are in agreement with the previous results chapter, and then performed the metabolic flux analysis, showing coherent results with previous metabolic metaphor analysis, supporting the growth rate hypothesis described in previous chapter, then showing the impact of dilution rate on metabolic flux distribution that is in agreement with previous studies, and finally checking and validating the metabolic flux distribution for redox cofactor balance and energetic requirements. So finally, the last chapter of results is redox engineering by ectopic overexpression of an NADH kinase in recombinant Picha pastoris. First of all, I would like to introduce one of those cell engineering strategies that can be applied in order to enhance the recombinant protein production. That is cultivation in hypoxic conditions. That means reducing the oxygen availability. In this case, despite Picha pastoris is a Crabtree negative organism, it turns its metabolism toward respirofermentative. In this case, the oxygen limits the oxidative phosphorylation, and NADH is accumulated. Indeed, in a previous thesis, it was estimated that the NADH to NAD ratio increases by almost 20-fold when comparing hypoxic with normoxic conditions. In this case, this NADH can be regenerated by giving ethanol or erythritol. In addition, it is also reported that in hypoxic conditions, there's a transcriptional regulation of the glycolysis. Therefore, if we express our recombinant gene under the control of a glycolytic promoter, we will have our recombinant protein production enhanced. In addition, I would also like to highlight the importance of NADPH in recombinant protein production. It has been widely reported the correlation between NADPH availability and recombinant protein production yields. In addition, NADPH plays a key role in amino acid biosynthesis as well as in protein folding and ER oxidative stress. So the question now is there any way of converting this NADPH to this NADH that is in excess to NADPH? In this chapter, we tried one of these strategies and used for studying the effect of redox cofactor balance perturbation on cell physiology and recombinant protein production. For doing that, we wanted to increase the NADPH availability, especially in hypoxic conditions. In addition, we expect to enhance the recombinant protein production yields. And finally, we use the genome-scale metabolic model for assisting the metabolic interpretation of the observed experimental data. Here's the graphical experimental procedure. First of all, we selected the POS5 gene that is encoding for NADH kinase from Saccharomyces cerevisiae. So it is converting NADH to NADPH by consuming ATP. This kinase or this gene is commonly expressed in the mitochondria into Saccharomyces cerevisiae, but we also try to clone it for cytosolic expression. So with this, we screen for the generated clones and analyze its gene copy number, and then perform some chemostat cultivations in different conditions, that is using glycerol, glucose, and glucose in hypoxic conditions. Finally, we use the model in order to interpret the resulting physiological profile. Here's a result of the screening. As you can see here, the fold change in fat productivity with respect to the control strain. As you can see here, we have a population that has around 2.6 fold change with respect to the control strain. So the fat productivity has increased more than the double. When we check this population for the gene copy number integrated, so the number of copies of POS5 integrated, we could observe that this population had 2 copies of the cytosolic POS5 integration. The other populations had just 1 copy of the mitochondrial and the cytosolic POS5 integrated. So for performing the chemostat cultivations, we use these 4 strains, that is the control strain, CPOS5, including 1 copy of the cytosolic POS5, 2 CPOS5, 2 copies of the cytosolic POS5, and MPOS5, that was the mitochondrial expression of POS5. During the cultivations, we took some samples for analyzing the NADPH to NADP ratios. For example, if you look here into the hypoxic graph, we can see here the representation of the 4 strains, the control strain CPOS5 to CPOS5 and MPOS5. Then in the radial axis, we see the NADPH to NADP ratio. As you can see here, for example, CPOS5 had an increase in the NADPH to NADP ratio. In addition to CPOS5, this increase has been even larger, showing more than the double of NADPH availability with comparison to the control strain. So meaning that there's a correlation between the gene copy number and then the observed NADPH-NADP ratio, or relating to the enzyme activity. Now, if you look to this shape is similar than in the other graphs, so the behavior is also more or less the same. Nevertheless, in hypoxic conditions, we see that these ratios are enhanced, and so meaning that the activity of POS5 is boosted in hypoxic condition. By looking to the macroscopic growing parameters, for example, we didn't observe any dramatic impact of the expression of POS5. Nevertheless, we can see some tendencies. In the case of oxygen consumption rate, we can see here that there's an increase of this oxygen uptake rate in the recombinant strains. By looking to the CO2 production rate in all the conditions tested, we see an increase of the CO2 production rate. Nevertheless, in hypoxic conditions, the mitochondrial strain show a reduction in the CO2 production rate. Regarding the byproduct profile, we can see that the cytosolic expression of POS5 showed similar or higher values of both arabitol and ethanol. In the case of the mitochondrial strain, in hypoxic conditions, it is shown a reduction of the ethanol production rate. Finally, and maybe most important, that it was one of our objectives in this chapter, is to look into the fat productivity. Here's a representation of the fold change in fat productivity with respect to the control strain. Here you can see that the strain that incorporates 2 copies of the cytosolic POS5 show the higher productivities in all the conditions tested. In addition, the one that incorporates just a copy showed similar or higher values of the fat productivity, while the mitochondrial strain showed no significant improvement. So by trying to integrate this information and also using the genome scale metabolic model, we first want to highlight that in previous studies it has been already reported the correlation between NADPH availability and recombinant protein production. For example, in a recent study, they tried to overexpress the oxidative branch of the pentose phosphate pathway, that is the main NADPH generation pathway in the cytosol. So, in these strains that has the NADPH generation increase, they also observe an increase in recombinant protein production. We also check with the model that there's a correlation of fat production with NADPH turnover. Finally, by integrating data from NADPH availability with the fat productivity, we can see that the strain that has the higher NADPH availability or NADPH to NADP ratio is also the one that has higher recombinant protein productivities. So now, to zoom into the metabolism, we perform this with our metabolic model, and we try to predict or try to reproduce this experimental data by increasing the flux through the NADH kinase reaction. So here, in the case of the cytosolic POS5, in blue, it is represented a decrease in the fluxes, while in red, it is an increase of these fluxes. So looking into the cytosolic POS5, we can see first that we predict a reduction in the oxidative pentose phosphate flux. As I said before, the pentose phosphate pathway is the main NADPH generation pathway. Therefore, if we have alternative reaction generating NADPH, there's no need of generating this NADPH in the pentose phosphate pathway. In addition, it has been reported in Saccharomyces cerevisiae that an excess or high concentrations of NADPH can inhibit the first step in this pathway. So it is coherent with these previous observations. In addition, POS5, apart from generating NADPH, it consumes NADH as well as ATP. Therefore, oxidative phosphorylation has to be more active in order to provide this additional ATP. Nevertheless, as we also are consuming NADH, there's no NADH available for feeding the electron transport chain, and therefore alternative electron transfer mechanism has to be activated. These alternative methods are less efficient, and therefore more oxygen is required. These predictions are also in agreement with the observed experimental results from chemostat data, in which we observe an increase in oxygen consumption. Finally, in hypoxic conditions, there's no way of increasing the oxygen consumption, as we are limiting in our cultivations. Therefore, as I said, in a hypoxic condition, there's an accumulation of NADH that can be used for generating ethanol, and we observe an increase in ethanol production. And this NADH can also be converted to NADPH by POS5. At the same time, this NADPH can be regenerated by producing arabitol, and we also observe an increase of arabitol production in our experimental data. And finally, it also can be used for fat biosynthesis. And also, again, as we can see in the experimental results, in this case for the cytosolic POS5, we observe increased fat production yields. Regarding the mitochondrial POS5, we have also the same. There's an ATP and NADH strains, but this time these strains are in the mitochondria. So NADH is consumed for giving NADPH. As we are consuming ATP and NADH, the oxidative phosphorylation gets enhanced. And so, as well as we observe in our chemostat data, there's an increase in oxygen consumption. As we are consuming NADH, it has to be alternative pathways for generating such NADPH. That could be the activation of malic enzyme or an aplerotic feed of TCA. Here. Finally, we predict that in these conditions, we have an enhancement in the pentose phosphate pathway activity that is in correlation with higher biomass yields observed in the mitochondrial strains. Finally, in the case of POS5, in the case of this mitochondrial strain in hypoxic conditions, part of this NADH would be transported via mitochondrial redox shuttles and then used for more NADPH generation. Therefore, if there's less NADPH available, the ethanol production will be also reduced, as well as in our experimental data from chemostats. So finally, as a summary of this chapter, we generate 3 different strains. That is the one copy of POS5, 2 copies of POS5 expressed in the cytosol, and the mitochondrial POS5. Then these strains incorporate the NADH kinase that has an impact on cell physiology. Cells expressing POS5 are more sensitive to oxygen. We observe that in hypoxic conditions, the effect of POS5 is boosted. In addition, the cytosolic overexpression of POS5 showed greater productivities of fat, and finally that there's a correlation of gene dosage and the observed phenotypes. Finally, our calculations using the model demonstrate coherent results with the experimental data from chemostat cultivations. So finally, as general summary, we merged the pre-existing models into a single one that we extended especially for Picha-specific metabolic pathways, and then formulate a new biomass equation. In addition, in the second chapter of the results, we showed how we extend this model by incorporating new biomass equations representing growth on glycerol and on methanol, realizing a new version of the model. Finally, in the last chapter, we showed some 2 different applications of this model. That was its reduction to a core model. Then we performed chemostat cultivations using labeled glycerol to perform metabolic flux analysis. Finally, in the last chapter of results, we used this model to try to give a better explanation, a possible better explanation of the observed phenotype. Finally, I have to say that this model has been used in the context of an European project by a company who tried to develop or try to predict new targets for model-based new targets for high value-added chemical production. So now, if the members of the examining board considers, I will read the conclusions. No? Otherwise, I would like to thank all the people from the department, as well as the research group, as well as the people from the Technical University of Denmark, especially Mary and Anders, and of course the funding institutions.
Okay, thank you very much.
Okay.
Then we will proceed then to discuss with Marius several aspects of your presentations, and the first to take the floor is Professor Val from the Delft University. I apologize, I cannot present the members of the jury. Isabel Rocha is from the Universidade do Minho. Alois Val is from the Delft University, and I am emeritus professor from this university. So, is that what? Yeah.
Okay, Marius, thanks a lot for the presentation, and yeah, it's been a pleasure to read your thesis. Very extensive work. So So I think the community can really benefit from having a curated metabolic model for Picha pastoris and put efforts into putting the right numbers into these models. So I can imagine that there has been really a lot of work and a lot of thinking. So as I understand, basically your final goal is to actually engineer now towards protein production. So you want to make this antibody fab, and your model then helps you in developing that strain. So I very much agree that modeling will help you in understanding how to develop it. On the other hand, you could say that microbial metabolism is kind of super optimized for making proteins, right? So this is just another fraction of the biomass. So in how far do we still have possibilities to actually optimize that? And then it comes to the next question. If you look into your current productivities of the protein, you're in the range of micrograms per gram. By biomass, you have half a gram per gram of protein in there. So why this big discrepancy, and how far do you think do we still have possibilities to actually engineer a system that has been evolved for years? And then maybe related to that, if you now look from the bigger perspective, so you claim that there are energetic and redox balance problems. So if you now take the model and actually calculate what fraction of the turnover goes into your product, how relevant is it then?
Okay, so regarding the first question, we know that microbial cells are really optimized for reproducing itself, so growing as fast as possible and then producing lots of proteins. Nevertheless, we know that when we are trying to engineer a strain, maybe we are affecting its global fitness of the cell, so we know that it would be possibly affected, but counterbalancing for example, the recombinant protein production. So we have to get an equilibrium between, okay, we are damaging or making that the cell is not growing that good, maybe, for our interest. So if, for example, if we are feeding with 100 moles of substrate, and usually these 47 are going to form the biomass, but we want to increase our recombinant protein, for instance, so it's obvious that these moles that were going to the biomass will go to, for example, to the protein. So we are affecting the overall growth of the cell. We know, and we had to achieve a balance between how we are affecting and then producing. So we know that maybe there's no way of increasing the recombinant protein production by keeping all the cell capabilities available. So we can maybe sacrifice one of these.
Slower, right?
Yeah. For example, some of the algorithms for trying to find some genetic targets, maybe you have to specify which percentage of growth rate you want to keep. So it assumes that you are reducing your cell fitness in order to enhance other properties. The same for metabolite production. If cell keeps its metabolic flux distribution in an optimal way, if you want to redirect your metabolic fluxes towards another direction, it wouldn't be an optimal cell physiology. So we know, and we had to try these gene targets that has a less effect on this cell physiology. So yeah, I know that it's quite difficult to improve a thing that has already been optimized, but we know that we have to sacrifice this optimal growth in order to get our protein, for example.
No, fully agree. I mean, you will have lower growth rates if you make some for the cell growth useless protein, but for us very interesting protein. So can you now put numbers? You have now this model. So how much of the protein of the NADPH and ATP turnover goes into your protein production?
Well, I think I don't have exactly the numbers, but in a slide here I had more or less a correlation.
Yeah, I could also in your book not find the number, but I found one figure where I could calculate it from, and then it was 0.05% of the time. Yeah, exactly this figure.
For example, here,
Yeah.
you can see here that, for example, is...
Yeah, so the difference between maximum production and zero production divided by this 1.07 something. So I came to 0.05%, which seems still quite a low value.
Yeah, well, I have to say that this is just the NADPH turnover for synthesizing amino acids, and well, the related for the required ATP for generating the protein. But we haven't taken into account all these redox equivalents needed for protein folding and secretion processes. So they are not still represented in the model, and this can be used in further model versions to incorporate, for example, the energetic requirements on
yeah, but that's
secretion.
energetic, right? That's not redox. So
well, but indeed we need NADPH, for example, for protein folding. So NADPH is involved in protein folding.
So where do the electrons then go in the end?
Sorry?
Where do the electrons then go from NADPH?
From NADPH, electrons, sorry, just a second. Well, more or less, there's a figure in the introduction that more or less represents this, what's going on with NADPH here.
Yeah, so in the end it goes to oxygen, I assume. Huh?
Yeah, sorry.
Yeah.
Here we have. Here is commonly how disulfide bonds are formed in the ER.
Yeah.
So there's the formation of these disulfide bonds, then there's a redox chain that ends with NADPH to NADP that are transferred to oxygen and other oxygen reactive species. So that's the oxidative stress in the ER, and the role of NADPH trying to alleviate this.
And can you put a number on that amount?
No, we haven't tried, or no, sorry.
But you expect that to be much more than the 0.05% of the turnover?
Yeah, maybe, but I'm not sure. I haven't looked into this.
Okay, and then going also back to the strategy, so now you say that it's redox limited, so then basically you enforce the redox by limiting the redox sink, the oxygen. But when you do that, you actually run into the other challenge that actually your energy gets limiting, because your ATP generation goes down significantly.
Yeah.
And so can you give a postulation on where's the optimum then, and how could you maybe use your model to actually find that optimum? So if you now really want to design the perfect FAB producing organism.
Yeah, of course. For example, here we can correlate the NADP H kinase activity. So if we had the strain with just one copy and then the strain with one copy, we suppose that we can perform strain with more copies. Therefore, by using the model, when indeed in the simulations we perform, we try to just simulate what happens when we put I don't know, for example, millimole per hour and gram of diacetyl weight in the model of this NAD of this post-5 reaction. Then we increase this flux in order to see how it evolves. And so the pathways, the increase and decrease pathways are explaining the tendency of increasing this flux. So at one point, we can say that the biomass yields decrease a lot. So we have to really, we can more or less estimate which is the point with the optimal fat production, but the biomass is not affected. in order to perform this with the model, we also can put some constraints in the model. That's, for example, ensuring.
Would your model predict oxygen-limited conditions as favorable conditions?
I think
If you optimize
not actually. What is predicting is that this excess of NADPH that we are generating can be used for fat production. So it's not really. It's actually only a stoichiometric balancing. So we don't know exactly if these conditions would be really optimal. It would have to take into account several other aspects such as gene profile or other kind of regulations maybe that are still not taken into account into the model.
So I understand correctly that then, let's say, the kinetic aspects might be more important than the stoichiometric?
Yeah, and other kinds of regulations and so on, but I think that what really does the model, or that in hypoxic conditions we predict an enhancement of the fat production, is due to that in the fat production there's a regeneration of such cofactors that are reduced. So maybe that would be a possible explanation of also that we are predicting high fat productivities in this case. So in fact, by producing fat, we need these reduced cofactors. So I think that's the reason, the main reason of the increase in fat production that we also observe in hypoxic condition, just stoichiometric.
Yeah. Maybe one approach that one could think of to actually increase yields would be to not increase NADPH production, but actually reduce NADPH consumption. So if we would be able to convert protein production from NADPH dependency to NADH dependency, I think it's actually only one gene that you really need to change, glutamate dehydrogenase. So has that been tested in Picha, and what was the effect on that?
I'm not sure if it has been tested in Picha. I'm I don't know, maybe it has, but now I'm not sure about it. But yes, changing the cofactor specificity could be a good way of playing or trying to modulate this cofactor balance.
So then a very technical question. So this POS5, as far as I understand, it takes NADH, phosphorylates, and you get NADPH.
Yes.
Then you use this NADPH, you have NADP. What's happening then? So how do you get the regeneration? So how does it get to NAD and then back to NADH? Or
okay.
So how does this change the turnover?
Yeah, I think that usually cells have mechanism for dephosphorylating the NADPH, but not a common mechanism for it has a mitochondrial kinase also in Picha, but very low expressed. So there's a way going toward the NADPH phosphorylation, but I think that the most common and more expressed is the dephosphorylation of NADPH. In addition, I'm not sure because we haven't tested this, but some enzyme specificity could be changed, so there's maybe no need of that amount of NADH, and it can turn its activity by using NADH.
In your model, it's then dephosphorylated, and it's regenerated then NAD to NADH, and then again phosphorylated. What happens?
Yeah, it has to be balanced. So, but I think that mainly the NADPH. One of the possible pathways is about the hydrofolate and conversions with this.
But if you just look now into your fitting sort of the experiment, how much flux did this POS5 then carry? So how active was it? Because in your presentation you said that this had now an influence on the oxygen uptake.
Yeah.
So this basically must carry an enormous flux.
Indeed, we haven't tested actually to reproduce exactly the experimental data observed. What we tried with the model is just a conceptual proof of which is the impact of expressing both in cytosol or mitochondrially this POS5. So we haven't tried to exactly reproduce to find the exact flux of any this POS5. So maybe in further studies we can just check. For example, we haven't published this yet, so maybe it can be incorporated before publishing. Yeah.
Okay then. Do I still have time for two questions? Yeah? Okay. So you describe your model as a consensus model.
Yep.
So I was wondering, consensus with whom?
Well, with the previous models. Yeah.
Okay. Because the Latin definition basically says it's a model agreed upon, right?
Yeah.
So do the previous models agree upon that model then, or?
Well, indeed, it's a consensus in the sense of just being all the three together, so they are like.
So it's more a community.
Yeah, it's a merge model.
Yeah.
That takes the best of each one, so it's like consensus. One of the main objectives I think for Picha community would be to really perform like a consensus, like we found on the case of Saccharomyces, human, and other most common organisms, in that the whole community just meet in order to provide new versions, and that everyone on this community is agree on that, and so they are continuously releasing new versions from this community perspective. So it would be really interesting for Picha to perform this in the same direction, and now really provide really consensus. I mean consensus with the Picha community models. Yeah. So maybe it's the first step by merging the previous models and then providing it for whoever wants to.
So the biggest step in terms of taking the best from the worlds and adding something to it is this complex one that you implemented?
Sure.
So if you compare now the non-consensus that was there before and what you now have, it's the complex one that you added to the model, right? As an important aspect.
Yeah.
And Arabitol production that was also somehow not present there. And I understand a couple of other inconsistencies. So now if you look into this complex one, so you have an NADH dependent complex one, you have even also an NADPH dependent complex one, and you claim that you now reach a stoichiometry of 4 protons being pumped out. So if you do the sum, you come to quite a high ATP yield of 32 ATPs for complete combustion of glucose to NADH and using that through the oxidative phosphorylation.
Yeah.
So I was wondering in how far does that now really compare with the experimental data, and because at the same time you play around with the growth associated maintenance and the non-growth associated maintenance. But I understand that Picha can also grow anaerobically, so actually you could also...
Completely anaerobic? No, I think...
Or with other electron acceptors?
I think that in completely anaerobiosis is not able. So maybe just reduce oxygen availability. For example, 0% of dissolved oxygen Picha can grow, but if you are not supplying completely anaerobic, so it's no, I think it can't.
Okay, but nevertheless, so you get then a certain PO ratio in the end, and PO ratios have also been estimated previously, but just based on kind of black box stoichiometries and kinetics. So what is actually your final PO ratio if you look now back in your model? Is the complex one active in all the cases, and in what ratio does it actually end up then?
I think we suppose that mainly is just generating by the classical with complex, all the complex one being active. And so I cannot remember the numbers, but I think, I would have to check. I don't remember.
On page 68, you give a number that a little bit confused me, so maybe you can elaborate on that one. There you say that you have 2.5 electrons per ATP. I think it should be ATP per electrons, and then not per 1 electron, but 2 electrons. So is your PO ratio then 2.5, or is it the 0.8, what you wrote there with the 2.5 electrons per ATP?
So I think, let me have a look. So
Yeah.
I don't remember by heart now.
Page 68 is where I had it. But 68 in terms of, let's see. No, this is the real, so including the previous pages, so I think there's some offset in it. 13 pages offset, yeah. So it would be 45 then.
45. So do you have the real page number? So
Yeah, I'm looking it up now.
Okay.
55.
Okay.
And then on the top of 55, just before the new heading, other reviewed pathways, resulting in a final maximum theoretical stoichiometry of 2.5 electrons per ATP.
Yeah.
So one NADH has 2 electrons, or an oxygen has 2 electrons. So then you would get a PO ratio of 0.8, turn it around,
Yeah.
which seems an extremely low value. 2.5 would be quite an okay value,
Okay.
but as PO ratio. So how
Okay,
did exactly manage this value?
Yeah, maybe I would have to really check the numbers here, and maybe there's a mistake on writing this sentence. I'm not sure, and well, I think we would have to check in order to validate these numbers. No, sorry, but I cannot answer with more detail. I think it would require to revise all the pathways we use for building that.
Because I mean, if that is true, that you get this 2.5 as a PO ratio, I think the Saccharomyces community would love to have these enzymes at hand and improve their organisms. Because, then you get 16 ATP per glucose, not 32.
Yeah.
It's quite a difference, and you got quite improved yield, so...
Yeah, okay. The only thing that I have to say that it could also be taken into account, and they make reduce this value, so that would make this less efficient, is that there's not taking into account any proton leakage or something like that. So maybe by taking this into account into the model, we can readjust to more, not theoretical values, but more realistic ones.
I think complex 1 in Saccharomyces cerevisiae, we have to talk about this, Paul. Okay, I think these were my main questions. Thanks a lot.
It's always a pleasure talking with the people about stoichiometry and thermodynamics. Thank you so much. No.
So I'd like to start by thanking the university and the department, and of course especially Paul for inviting me to be here, and also to congratulate Marius for reaching this stage, and also for writing this thesis. It was very nice to read. I think it's overall well written. Also, I'd like to point as positive aspects the rigorous methodology in most of the work that was performed, both in the lab and theoretically. I think it's not obvious for people who read it the amount of work that is involved. Only if you are familiar with characterizing biomass in so many different samples, also performing C13 labeling experiments, GCMS, and so on and so forth. So I think this is perhaps not totally clear if you haven't done this work previously. So I will start with one generic question, and then I'll go for more details, and I will end up with more discussion or wise questions. The first generic question, you mentioned, I think in the introduction, but I guess also along the text, that Picha pastoris is more and more seen as a platform for biorefinery. And I'd like you to comment on the fact that, well, for protein production, I think it's clearly an industry has recognized it as a very good host. But what about, well, when I'm talking about biorefinery, in many cases you're talking about metabolites, smaller compounds. Do you think that Picha pastoris is a good platform for producing compounds for industrial biotechnology, and what compounds would you think make more sense?
Okay, so first of all, as one of the main advantages of Picha is that it's an eukaryotic host, so you can differentiate the expression of a gene in compartments, and this may be interesting for producing certain compounds in order not to mix these compounds or these precursors with other cell components, and that will affect maybe the cell physiology. In addition, by comparing different eukaryotic hosts, for example, in the case of Saccharomyces, it has a very low growth on glycerol. And glycerol is one of the main compounds used, in this case as biorefinery or a substrate for producing high value-added compounds. So we can use Picha for producing compounds that use glycerol, for example, or that are by modifying glycerol. And here's in the presentation is an example that is, for example, the 1,2-PTO that basically modifies the glycerol skeleton by adding some alcohols in there. It also can be, well, used for
but
this.
For which cases do you think there is enough proof of concept and comparing with other organisms, like, for example, Saccharomyces or E. coli?
Well, I think that actually Picha has been started to be used in this way, so there's no an extensive body of knowledge of using Picha for producing high value-added chemicals. But there are few examples, and knowing the properties of Picha, so it is pointing that in the future years we will find more data on this. We have characterized Picha, and we have lots of tools for genetic engineering Picha that can be easily adapted for expressing other genes and other enzymes. So for now, maybe we don't have extensive data, but for sure we will see in the following years. At least our group will try to provide some of these new examples, hopefully successfully.
I totally agree with the part of the substrate consumption. I think it's promising in that aspect. I'm not so sure about the production part because even Saccharomyces compared with bacteria, it loses regarding most of the compounds. So it's not clear. It would be for very specific compounds, and you would have to analyze the pathways and really prove that it makes sense because they naturally already have a higher flux. Because otherwise, I'm not so sure, because these organisms have evolved a lot to produce biomass, and it's probably not so easy to couple biomass with a product.
So maybe Picha won't be the default organism for chemicals production, but in a specific case regarding specific compounds, it can be used as a...
Yeah. So that's why I don't think I agree with the platform term, but I wanted to explain this a bit. So I now divided more the topics in terms of the modeling part, and then I'll go for what I call the biomass topic, which relates with different chapters. I have some doubts regarding the model, and first I would like to acknowledge the work that you have done We have also tried to make model integration and reconciliation, and it's not easy. And I also worked with some of these models, and I'm very conscious on the limitations that they have. I would like first to start from the beginning, which was the fact that you had two strains, right? Mm-hmm. And these strains have 93% identity, and you pass over this number without going into the detail. My question is, first of all, if you know if anybody has done any direct comparison of the genes, like for example doing a bidirectional best hits analysis to see which are the genes that are really present in both, and use this information for, and if this has been done, if you could use this information for the model, or even if you have done it.
Okay. First of all, I haven't compared this information from the strains, but nevertheless, based on the available bibliography, it was reported that despite this difference in the protein or in the sequence, this 97% of
wasn't it 93?
93, yeah. That despite this difference, there are no major metabolic difference, or the metabolic behavior of the strains is not reported to be different. It doesn't mean that they don't behave differently in certain conditions, but at least there are no major differences between the strains. Of course, maybe these calculated energetic parameters or specific biomass composition can change more or less, but I think that it can have some effects on the internal metabolic flux distribution, obviously, but
so you would expect that the differences are not in the metabolic genes, but they are rather in?
Well, maybe some specific mutations on some metabolic genes, and maybe yes, the activity of an enzyme could be different, or the affinity could also be different, but the global metabolic network topology would be quite the same. I haven't checked it, it's just assuming that, but I don't have noticed, or I didn't notice that nobody has performed this study.
Yeah,
it would be interesting now that there's another refined genome sequence for Picha, in order to compare all these genes. There's a platform available, I think it's pichagenome.org, in which you can compare different Picha strains in order to get the gene homology, for example. So it would be interesting, for example, to provide a model that has exactly the correlation, or you can adapt the model for each strain for performing calculation.
Yeah, exactly. I think this can always be done, and it's not. It's something that there are bioinformatics tools to do it, and then you can have strain-specific models if it makes sense. Because I agree that you're not necessarily expecting many changes in the genes that codify for enzymes. You expect more conservation on those and less conservation in regulatory genes. But nevertheless, there might be some genes that are not present in one strain and are present in the other. Another related aspect is regarding the annotation of the genome. And I think I saw some sentence, and at least I didn't see anything else regarding annotation. Because you seem to mention that the annotation available was poor. Is it still the case, or did you only use the annotation that was available, or did you use other tools like this database that is now available?
Basically, we used the annotations that were in the previous models.
Okay.
And in case of inconsistency between among the models, we then check for the gene of each gene, and with several databases, not only just this one that compared the genome, but also comparing to this genome, another fungi ensemble, in order to check which are the possible candidates or in which organism it could be described in a more accurate way. For example, most of the organism is like inferred from, and then you don't have the certainty of this gene. So it could be this, but maybe somebody characterized or misannotate this, and so you get this information propagated.
But I imagine that you have performed some blasts in several things. So you actually did some annotation yourself.
Performed by some of these platforms.
Yeah, so you actually did some annotation. Okay, that was not clear, and I think it's still not clear in the text, because you actually did some annotation, and probably not in a systematic way, but in the genes for which you had some
divergences or discrepancies between the models we performed. But this was not the major work during the reconstruction.
If you had to start doing it now, would you start by doing genome annotation, re-annotation, or?
If there were, if we didn't have, for example, this new genome sequence that is high quality genome, maybe yes, it will be a good idea, or even it can be performed like doing re-annotation of this genome. So now with available bioinformatic tools, we can easily perform that. But if it is performed, it has to be a completely an extensive manual operation of that. Otherwise, we will propagate the same errors that we already have in the same model. So an extensive comparison, but not only comparison with other organisms, because we will have also misannotation or false homologs. So there's more a manual curation by looking also into the bibliography and so on. So it would be an interesting point. But in addition, there's a lot of work done in the previous model. So there's work done there. So I think the best option would be to re-annotate, for example, a new genome, a high quality new genome, then looking what can we extract, which metabolic network can we develop from this genome, and then also use the information available from the other genomes in order to compare, because in the previous genomes there are people who spend hours in looking also into it, so we can take some information around this in order to refine, or in case we don't know which gene associate, we can look, okay, these people associate this reaction with a certain gene, and so we have maybe more clues in order to elucidate which is the correct gene coding relationship, or to define some pathways that you cannot infer from the direct observation of the genes. For example, it was the case of the sphingolipid metabolism in here, that okay, maybe the genes are correct, but that the pathway don't make sense. So you can take the information from the previous models also in order to perform this new one with the new re-annotation.
Yeah, but any annotation has to rely on existing information, and some pathways are not totally covered in the existing in existing databases. So that is a problem that we then not only propagate errors but propagate ignorance.
Yeah.
We don't know the function of many genes. I had several other questions, but I will. When you did the model reduction, I didn't see. I saw comparison of the results, but I didn't actually see a comparison of this new core model with the models that other people have been using for 13C MFA. Did you do it, or even in your group? I guess you were using another smaller model before. How does it compare? Are there inconsistencies, or is just like increasing the scope?
Well, apart from including all the metabolic pathways, because in the reduction you get more than just the central carbon metabolism. So biomass is more accurate representative. But we haven't compared with previous results. For example, we have here a study that just altered Picha using glycerol at different growth rates. Nevertheless, it was just applying the metaphor analysis, and we could only check is just these variations depending on the growth rate. But we haven't compared the overall performance. What we realized, for example, is that in previous models, some pathways were represented. For example, the glycerol uptake rate when performing the redox cofactor balance was not accurately represented, or represented in a different way than the one that we suggest. That is the first reduction and then a phosphorylation, and here we suggest a phosphorylation or a reduction, but I'm not sure now.
Yeah,
if
okay.
this
This is the type of things that I was hoping to see a comparison. Not, of course, I know that you really increase the scope, so it covers many more, but regarding what the other models were representing, was there anything wrong there that could affect the predictions?
Okay, so you mean comparing, for example,
the
adjustment using the other metabolic
not necessarily
network?
the results, only the structure of the network itself. Was there a reaction that was misconsidered there, like what you were saying, steps that were confused? I think that would be valuable to have this comparison for the community, because one thing is to say, okay, I have a much detailed model and it works. But people can still use the other one. Another thing is to say, well, look out, the other one has some mistakes or things that we didn't know at that time, and now that we have all this genome scale information we have. So I think it will be useful for the community. I'm not sure if you already published this part.
No.
So it's a suggestion. So jumping to the biomass part, I have only two short questions. Maybe starting at a certain stage, you say that you had essential cofactors as biomass components. I didn't see those in the annex, so I would have to go to the SPML, and honestly, I didn't go. So can you share not necessarily the list of all these cofactors that you included, but the criterion to include those in the.
Okay. We, apart from being present in previous models, just an equation representing the cofactors, when we try to evaluate the essential genes and the active pathways when growing, at first we realized that if we didn't include these cofactor equations, there were many important pathways for cofactor generation and, for example, NADH or some other specific cofactors that if we don't include these specific equations, they are never active.
Mm-hmm.
So we just included this series of...
Did you take it from any existing model?
From a previous model.
Okay.
Maybe it's not... I think it's an extensive list, but of course can be revised for including or not considering others. But we took this information from previous models.
And you checked that it made sense regarding the active pathways.
Yeah, so maybe there's not an accurate representation of the amounts that generated, but it's just to avoid pathway inactivity.
Yeah, we have actually developed a methodology to try to look for those, but for prokaryotic organisms. That's why I was curious, because to see how for eukaryotes you reach this list. But you are right, it affects a lot. And then afterwards you say, so when you are discussing the wrong or correct biomass composition regarding different carbon sources and even different growth rates, if in your case you didn't see a significant difference, but you mentioned that when having a wrong biomass composition, this may lead to wrong predictions, including essentiality predictions. Mm-hmm. Do you think that changing the proportion of protein in the biomass equation, or changing the proportion of lipids or carbohydrates, it affects the predictions regarding essentiality? Well,
just changing the proportion of these macromolecules, of course not.
Mm-hmm.
So
what would change essentiality?
For example, the specific composition on lipids well, amino acid profile not, because all the amino acids need to be synthesized. But here, one of the, I think that is the most effective on predicting these essential genes is the lipid profile. Indeed, we observe that the lipid profile, by comparing glycerol grown cells with methanol grown cells, is significantly changed, at least in some points. And I think that
but
some
what do you
of the
mean
lipid
profile? The relative composition of each lipid?
But I think that even one lipid species was not present in methanol.
Because that's what really changes essentiality, is the absence or presence of a given component, not the relative composition either of the macromolecules or of the building blocks.
Yeah, but in lipid analysis, we with the composition report, we realized that there was some species that were not present in one of the conditions. So, yeah, for example, the gene for this reaction would be affected.
Okay, so as a final question, because I think I'm already... As a final question, it's more of a discussion, open discussion. Although we don't necessarily discuss this into detail, I think you are aware that psychometric models are not necessarily a good representation of the type of phenomenon that you are studying and the type of phenomenon that you include in the introduction regarding all kinds of problems that happen during recombinant protein production. I don't know if you are aware of the what people now call ME models, or models that include protein production and protein secretion, which are more complex and complete models. How do you think these type of models would improve the predictions for this type of process, or do you think it's just a minor improvement and that we need other types of models, like for example dynamic models that would capture the dynamics of what happens during a stress response or something like that?
Well, I think that the more information we have, so the better predictions we can perform. So by including this new stuff, for example, the recombinant protein process, for example, as I said before, we can take into account this ATP age regeneration, for example, or some other requirements. The same for gene expression. We don't have information on gene expression, and maybe we have to constrain our model in order to reproduce a certain condition or gene regulation or thermodynamic regulations. Basically, in these models, I think they are starting being implemented, but they are not commonly used for genome scale models. But I think that the more information we have, the more accurate can we predict.
But not necessarily, right? The model is always a model. And like you showed, for example, for the C13 MFA, it wouldn't make sense to use the genome scale model that has more information to use as a platform for performing C13.
Of course, it always has to be taken into account the application you want.
Exactly. Because you will never have all the information that represents the process or whatever you are representing.
Thank you. So, first of all, I'm very happy to find the brilliant student I met in your third year and now presenting a so good dissertation. Then congratulations first of all. Then something general and some specific questions. I think that this way of presenting the thesis prepared by chapters to be published as such, complete with each bibliography, at the end has an effect of fragmentation. It's more difficult to follow
you present at the beginning what is the, let's say, the script, you know? But it makes more difficult to follow but this is not your decision, I imagine. And then the second is, it was supposed that the PhD was the personal research, a personal research, many years ago, still in humanities. But then, my first question is, in fact, what are your personal contribution to this work? So, because you are working in a team. My question is, do you perform all the experiments? Do you perform everything that is in this?
But for example,
because in fact the papers you are not the single. There are other people. These are your advisors.
yeah. For example, in chapter in the first and second chapter of results, that is developing the model and then extending, I perform the chemostat cultivations, as well as the biomass characterization with some additional help that perform some specific assays. And for example, in the first chapter, that was the model merging, I performed the comparison, and then when having these issues of how we represent this pathway, which would be the best representation of fatty acid metabolism compartmentalization and these Picha specific traits, we meet with my supervisor and then discuss which are the best options of representing these according to the information available. So in the second chapter, as I said, I performed the chemostat cultivations, and we also discussed the results in which direction is the growing profile, which are the possible explanations of the biomass dependent on stoichiometry. So we discussed all together. Regarding, for example, well, I performed the model formulation once we had the calculations done. Regarding the third chapter, so discussing with the supervisors which would be the most appropriate way of reducing the model, for example, which pathways or how we can perform such reduction, and the strategies or where to go to perform the experiment, the allowing experiment. And then finally analyzing the results, and of course they were there when the previous study on growing on glycerol was performed, so they could provide a lot of information regarding this study, so just comparing and discussing the results. And finally, well, in the last chapter is the one, the more collaborative one, that involve more and more people. So for example, by performing the chemostat cultivations, I think that they were like four different students or postdocs. Some of them are around here. And well, we performed all together this analysis that included the ADPH and the gene copy number characterization, but I performed the modelization part in this chapter.
Okay, then let's go to these chemostat experiments that you performed. I think it's in the slide 35, I'm not sure, it's in the page 89 of the documents.
I will try to put the figure here.
It's not exactly the same, but it is
More or less.
more or less is the same, yeah. First is, I think you are talking about the specific growth rate.
Yeah.
Not here. It's growth rate.
Yeah. Okay.
It's not the same. Then, how far are you from max?
Well, in the case of methanol, we perform cultivations up to 0.13, and we observe reactor washout, so meaning that the maximum growth rate was between 0.10 and 0.13. Approximately, we could estimate, not performing a very systematic study, but just looking how the biomass was decreasing in the reactor, and we estimate that it is really around 0.11. So the maximum growth rate for Picha growing in methanol would be at 0.11.
That means that you were performing very risky experiments.
In fact, we did want to check which was the maximum growth rate of methanol. So we performed the chemostat cultivations, and then the last condition we tried was this at 0.13, that we know that may be over the maximal growth rate. So we wanted to check how far can we go with the dilution rate. Yeah. And in the case of glycerol, we tested more dilution rates. We tested up to 0.32, more or less. But we didn't include it here because it required additional oxygen supply. And so we just wanted to do with common air.
Yeah. Then, if you look at the table, I'm sorry, 5.2 of your presentation and the thesis. This one, okay. My first question is, what is the explanation for the fantastic disappearance of degrees of freedom?
Well, because this just was the same model that we use here, and we just give more reactions due to this reversibility. We didn't evaluate it.
Yeah, but you have 77 reactions, 102 plus 9, 111 metabolites, and the degrees of freedom are 4.
We haven't calculated for this case because we assume that we would have more or less the same level.
How do you close the system then?
Well, by providing.
How can you find a solution?
Well, trying to find a solution that is closer to the experimental values, both the distribution and also the extracellular fluxes. In addition, I have to say that at least one of these extracellular fluxes was saved as constraint. Otherwise, we cannot calculate any fluxes. But it could be scalable, so we had to to establish one of these extracellular fluxes in order to be able to calculate all the others.
There is the optimization.
And there must be lots of conserved moieties in your model, otherwise it's impossible to get that. So, yeah, maybe you can state on that.
Sure.
Maybe you can say something about conserved moieties to answer this question.
No?
Yeah, so you must have conserved moieties in your model, otherwise the amount of metabolites cannot be that high. There are not enough reactions for the amount of metabolites.
Yeah, maybe.
So maybe you can state something on the conserved moiety.
Well, we have
There's
amount of
30 conserved metabolites. It's crazy.
yeah, I'm not sure about it now. So, in fact, there are some metabolites that are present in more than one compartment. So maybe it could explain this conservation of these metabolites.
Then, but look at the bottom line.
Yeah.
Mumax. Yeah. And if we refer to the previous slide, you were working at 0.16.
Yeah.
I hope that's much.
We just tried by setting a specific substrate uptake rate, and we tried, for example, the substrate I think that we just said this is. I mean, I can't remember the experimental substrate uptake rate, and so there's this value. I have to admit that the methodology by characterizing the biomass used in GTU was different than here. So maybe this quantification might explain these variations between this experimental growth rate and the observed experimental that was near 0.1. So, but nevertheless, I think that they are not quite deviate from the experimental dilution rates. So we tried, for example, giving the substrate uptake rate at, let's say that 0.1, that really was 0.90.
But Marius, one thing is the experimental dilution rate, and the other thing is Mumax, that is a microorganism characteristic.
Okay, so here is the mu max refer to the it's not actually the mu max. It's just the biomass, the specific biomass production when giving this amount that we gave to the model as substrate.
Yeah.
So it's not the mu max.
Then, you know, I like figures more than tables. and then if we go to the here is the. So you have to use it in your presentation.
I have all the figures.
It's in the page 101 of the S42.
Oops, sorry.
Yeah, are you happy with this? With your model?
Well, I'm happy that we could get the reason why the model is not predicting accurately, because there's a reason that is decreasing the energy maintenance at lower growth rate. So if we want to really try to predict or use the model in lower growth rates, we had to better calibrate or characterize the Picha pastoris growth on such growth rate in order to establish the proper energetic requirements. So, as it didn't follow linearity in the lower growth rate, so the commonly used energy requirements for glycerol is not the proper one. So, as we validated for a range of conditions, we know that our model can accurately predict the results in such range of conditions. So going at a lower dilution rates or even higher, we are not sure that our model is able to reproduce the experimental data. So in this case, we realized that there was a difference in the required energy requirements, so that we had to reduce to a new value for if we want to accurately describe the growth profile in this growth rate. So, of course, it would be better that automatically our model shifts this requirement these requirements, but then, well, of course, that should be ways in order to perform that when model predicts to be lower to a certain dilution rate, it changes the boundaries for the maintenance reactions to set the specific new requirement value.
Mm-hmm. Well, by the way, what does mean IMT?
Well, commonly, usually for the nomenclature of the models, it is commonly used the names of the first authors that developed these models. So MT corresponds to my initials. And then 2026 is not my birthday. It's the number of genes included in the reconstruction. In fact, previous models, not all, but some of them show, well, here for example, this one that is ILC is Luis Caspeta. So I think this one refers to the laboratory in which was developed, and this one is just Picha pastoris. But we can find many examples of other models that include it.
And the I is because you use an iPad? Or?
And no, all of them is like in silico, yeah. But then you can use in an iPad.
Well, yeah, that's in order to finish. It is in fact, I think, reading the introduction, I get the impression that any engineered microorganism is better than a wild one.
It depends on what do you want to use it for. For example, if you want just for growing, you better pick a wild type one. And for example, I don't know, it depends on the application.
No,
you cannot use engineered organism for wine production.
Yeah, no, I mean that, well, the idea is, my observation is that it is not taken into account that when you are working with that engineered microorganism, the facilities, the industry must be, is much more expensive if you are using a wild and grass microorganism as it is Picha pastoris. Okay, thank you. Any more questions? Then, so any of the present holding a PhD from the Autonomous University of Barcelona want to make a question? Then the session is temporarily closed.
Las cegatas del fondo.